Second generation sequencing, it's also known as next generation sequencing. It's the high throughput technology. It is used rapidly and cost-effectively sequencing of DNA or RNA. It is more advanced than the single sequencing or first generation sequencing. It's called revolutionizing of genomic research. We can say it's have a high impact on the genomic research. So we have different second generation techniques. So the most useful and dominant is Illumina sequencing. It's the workflow of Illumina sequencing. First we get sample, okay? From the sample we extract DNA. After that we start library preparation. DNA is fragmented up to 200 base layer up to 500 base pair. We add adapter, flow cell start to binding site, sequencing primer binding site, and also index barcode for multiplexing. And second step, clustering generation bridge amplification occurs. Each DNA fragment bind to the flow cell. They bend and form the bridge-like structure to the neighbor oligo. Moreover, PCR forms clonal cluster. Millions of clusters form on the flow cells. And third step, sequencing by synthesis. R4 nucleotide added at once to the each fluorescently labeled with different colors. It's incorporated when fluorescence emits signal. The camera record the base. How it record the signal is on the base of color. Okay, so on the base of color, it detect the nucleotide, like we have discussed before. That each nucleotide have individual color, and on the color base, it sequencing. Blocking group removed, cycle repeats. What we get in the fourth step is output. We get FASTA-Q file that contain a read sequence and quality code that is read. Second generation sequencing have multiple roles in the research field, and also it's have widely used in genomic sequencing. Genomic sequencing is to sequence the entire genome for research and clinical purpose. The second application of second generation sequencing is exome sequencing. Focus only on the coding region of the genome so that we identify mutations that are associated with the target disease. RNA sequencing that analyze gene expressions and transcriptome profile. Second generation have other various applications too on the base of their use. So this is the graphical visualization of second generation sequencing. DNA are extracted from the sample, fragmentation and adapter ligation performed, and also cluster growth performing PCR, bridge amplification. These are the bridge that amplifies, and also sequencing by synthesis method performed on the base of the fluorescent nucleotide are detected, on the base of which overlapping determines